Next speaker is Travis, I think, Travis Craddock. And he's here live, stepping up. And I'm not exactly sure what his, oh, here it is. Hallucinogenic compounds interact with microtubules to affect brain function. Adverse effect or putative treatment. Travis.
Thanks, Stuart. Here. Thank you. Hi. Can everyone hear me okay? All right. Stuart already gave you the introduction here. So first off, I want to say I'm from Southeastern University, which is in Fort Lauderdale, Florida. I left there to get out of the heat, which I accomplished. I left there to get away from hurricanes. Kind of failed with that. I work at the Institute for Neuroimmune Medicine based at NSU. Our goal is to try and find new treatments and diagnostics for complex chronic illnesses involving a neuroinflammatory component. So any neurodegenerative illnesses, as well as neuroinflammatory illnesses such as chronic fatigue syndrome, Gulf War illness, and now, following the pandemic, long COVID. This work is somewhat tangentially related to that, and it may open up avenues for new treatment avenues in things like PTSD or some of these neuroinflammatory illnesses. I'd like to acknowledge my team. The work I'm going to be talking about today has primarily been done by Isadora Dabro in the lower left-hand corner, with some of the modeling done by Caleb and Tatum. And I'd also like to thank the funding agencies that have funded a lot of this work over the years. And finally, I'd also like to thank the conference organizers. Thank you to Stuart, and thank you to Thomas for inviting us and having us here at this wonderful conference here in San Diego, Encinitas specifically. Okay, a brief overview. I want to talk to you about psychedelics. So we've heard from Arad. We talked briefly about anesthetics. I want to flip to another dimension that's kind of related to that. Psychedelics. I want to go and talk about the microtubule cytoskeleton. I want to define it a little bit better, describe it in a little bit more detail, and then talk about how do psychedelics potentially affect the microtubule cytoskeleton. That fourth point I will not be discussing. I removed that prior to the talk, but I didn't get it away from this overview slide. And then I'll finally talk about how psychedelics may affect what has been known as quantum mobility theory. So we heard a little bit about that from Arat, talking about energy migration in microtubules and how potentially anesthetics, they actually block energy migration in microtubules. But we'll talk theoretically about how psychedelics may interact with that same energy migration. So first off, a psychedelic, I think we all know what they are, but briefly, they're any sort of chemical compound that we humans can ingest to alter our sense of perception. So classically, these are chemicals that are known to cause trips. Primarily, when you think of this, you're usually thinking about a visual hallucination. However, psychedelics can also alter your perception of time, your perception of hearing, or any sort of sensory stimulus. It would be altered under these chemicals. Because they alter our sense of perception, they're altering the way that we view the world and how we consciously experience it. So like anesthetics, they are a natural probe for consciousness. Anesthetics, by far, I think, are the best natural probe for consciousness. It's something we can turn consciousness on and off with. Psychedelics are a little bit more difficult to handle because we really don't know how they're going to affect consciousness, but they do affect the way that we do perceive the world. Classically, it's understood that psychedelics primarily act by working on the serotonergic system, primarily interacting with the membrane receptor 5-HT, which is the serotonin receptor. In specific, 5-HT2A is one receptor that has been a prime location for psychedelic action. However, I'll mention it a little bit later, there is more experimental evidence that has recently come out by Varga et al. That talks about how psychedelics mediate their psychedelic effect not by the membrane 5-HT receptors, but by intracellular 5-HT receptors. So receptors for serotonin that are inside the cell rather than at the membrane surface. Psychedelics come in three broad main classes when you're talking chemically. They're the tryptamines, the lysergamides, and the phenylethylamines. And I'm going to be focusing on that last one in specific because that's the area that I've been working in. So phenylethylamines are a natural class of chemicals that are neurotransmitters. They're stimulant neurotransmitters. So we have natural endogenous phenylethylamines in our body. But there are also a class of exogenous or external phenylethylamines that we can ingest. They can be non-psychoactive, such as my asthma medication, albuterol, or phenylephrine, which is another chemical that's used in certain medications. Or they can be psychostimulant, like methamphetamine. You may be familiar with that from shows like Breaking Bad. Or they can be psychedelic, like mescaline, which you get from the cactus, or MDMA, which is an enactogen and a psychedelic, which is known to alter mood and perception. Specifically, I'm going to be talking about a small subgroup of psychedelic phenylethylamines known as synthetic phenylethylamines. So they have a base structure, which is that structure in the upper left corner there that has that ring structure with that little NH2 on the backbone there. That itself is phenylethylamine. Any sort of modification to that, any sort of substitution by adding a side chain or a chemical group onto the side is known as a substituted phenylethylamine. Like I mentioned, Varga et al. Had talked about how psychedelics in general could act at 5-HT2A. It is not known whether these substituted phenylethylamines act at those sites, but they do primarily act with that 5-HT2A system. But they can also target many other systems: dopaminergic systems, histaminergic systems, other systems that take advantage of those natural phenylethylamines. I wanted to look at something a little bit different where it's known that certain chemicals act, and that's the microtubule cytoskeleton. The reason I wanted to do that is back in the early 90s, a scientist, Van Wokerum, looked at the chemical structure of chemicals that are known to interact with the microtubule cytoskeleton, primarily colchicine, which you heard Arat allude to, as well as another microtubule inhibiting agent called the vinca alkaloids. And he looked at those and found that there was a strong structural resemblance between them and psychedelic compounds. So vinca alkaloid tends to look like your tryptamine compounds, which has a tryptamine group, or looks like tryptophan. It has an indole ring associated with it. And then there's the phenylethylamines, which looks like colchicine. So there in the upper panel there, you can see colchicine. In the bottom right of colchicine, you see that ring structure with the 3 O groups off of that. That, if you look at mescaline in the bottom right-hand corner, you can see that same ring structure with those 3 O groups off of that. And then right beside that, you see 2-phenylethylamine, which has that backbone, which forms mescaline. So all of these phenylethylamine compounds share that same sort of ring structure, and it's purported or hypothesized that because of that, they can act similar to places where colchicine would interact, like the microtubule cytoskeleton. Looking in the literature, we found evidence that mescaline can interact specifically with the microtubule cytoskeleton, and it causes it to inhibit microtubule and mitotic spindle formation. Whereas 2-phenylethylamine, that's shown in the bottom left-hand corner there, it actually promotes microtubule stabilization. So there's this kind of bidirectional role depending on what chemical side chains are attached to this phenylethylamine structure. There's also been shown, and this is interesting, and I want to talk to Stuart about this later, is that the 5-HT receptor system, especially the intracellular ones, have been shown to interact and modulate microtubules. So the way that the microtubules interact depends on these 5-HT receptor systems. So there is a natural functional role between the microtubule system as well as the serotonergic receptor system. So this suggests a biochemical basis for neuromodulation of phenylethylamines acting directly on the microtubule cytoskeleton. We've heard a lot about the microtubule cytoskeleton. I just want to talk more specifically about what it is. We're all talking about the brain. The brain we know is made up of brain cells or neurons. Neurons have this very distinct architecture where you have a cell body and these processes coming off of it. You have dendrites, which are these short ones around the cell body, and then you have the long axon, which sends signals to other neurons. Within that structure, you have these special filaments, which are microtubules, and they form in these bundles that give rise to the structure of that cell. So in other cells, we've heard that microtubules exist in your kidney cells and heart cells and skin cells, and that is true. However, only in neurons do they share this bundled architecture. In other cells, they're kind of dynamic and they're not stabilized with these microtubule-associated proteins. However, in neurons, they are in order to maintain the structure of the cell. Many diseases, Alzheimer's, Parkinson's, Huntington's disease, are associated with dysfunction of the microtubule cytoskeleton, as well as other diseases such as autism, bipolar disorder, dementia, depression. There's a whole book by Nancy Wolfe, Avner Priel, and Jack Tazinski, which discusses this. So what are microtubules themselves? They are polymers of the protein tubulin, which is shown up here in panel A. Tubulin is made up of two similar related proteins called alpha tubulin and beta tubulin, and they're always found together. And then that kind of peanut-shaped structure sticks end to end, shown over there in panel C, and forms these long strands of tubulin called protofilaments. And these protofilaments stick side to side and wrap around to form the hollow tube that is the microtubule. They play many roles, as discussed previously. I won't get into that, but that's generally the structure that we're talking about here. And altering that process of the way that those tubulin proteins stick to one another, as these tubulin proteins stick to one another, the microtubule grows in length, and that's called polymerization. And so what I'm going to be talking to you about is how these chemicals can modify that polymerization process. As we discussed before, there are numerous sites that you can target on tubulin to modify polymerization. Many of these are used in anti-cancer therapy, so paclitaxel primarily being a microtubule stabilizing agent. It prevents microtubules from falling apart. So when microtubules form the spindles that pull DNA apart during cell division, such as in cancer, if you have a microtubule stabilizing agent, they can form and grab the DNA, but then they cannot fall apart. And the cell gets stuck in mitosis, and it causes the cell to die. And that's why we use a microtubule stabilizing agent to treat cancer, because we want to kill those cancer cells. Other compounds, such as colchicine and vinca alkaloids, they prevent tubulins from sticking together, and microtubules can't even form in the first place, so you can't even get to the mitosis stage. However, most of these chemicals, Taxol, vinca alkaloids, colchicine, they're very poor at crossing the blood-brain barrier. It's not easy for them to get from your bloodstream into the brain. However, there have been anecdotal evidence that taking colchicine or vinca and directly injecting it into the brain will cause hallucinations. That doesn't necessarily mean that they act as hallucinogens. I'm sure that you could take any chemical and put it directly into the brain and cause something to happen, but it is anecdotally related to this kind of work here. But it does suggest that alterations of MT by certain chemicals like these may play a role in psychedelic experience. So what we did is we took a library of known phenylethylamines that are psychedelic. It is known, they've been characterized by many people. There's a book by the Shulgins called Phenylethylamines I Have Known and Loved. And they experiment with these phenylethylamines and they synthesize them themselves and then try them on themselves and record what happens. And so we can get a dose response curve kind of from their work. But what we did is we took the structure of those chemicals and we screened them against the colchicine binding site on tubulin. So it's a computer program that looks at all the different shapes and ways that those chemicals can fit within that binding pocket. It calculates out the energy of binding in that binding pocket, and the lower the energy, the lower the binding energy, the more strongly it binds within that pocket. And we compared that to the chemical colchicine, which is listed up there on the top. And colchicine binds with an energy of negative 10.8 kilocalories per mole, which is fairly strong binding. And then we ranked all of those phenylethylamines. We found that they all bound, some of them more strongly than others. And the ones that we were interested in, this is the top 10% of those compounds, they bind with a somewhat comparable energy, a little bit less than colchicine itself. So the first chemical there, DMB, MPP, binds with negative 9 kilocalories per mole. And then some of the other ones there, it kind of goes down to about negative 7.9. Now, what we wanted to do is we wanted to look at chemicals that we would know would maintain their chemical structure once they got into the bloodstream. So what tends to happen is when chemicals get into your bloodstream, a whole bunch of enzymes can act on them and your metabolism breaks them down, and it may turn them into another form of chemical. And then that other form of chemical may be the thing that is inducing some sort of hallucination. So we looked at these compounds, and the ones that are highlighted there are the ones that interact the least with that metabolic system of breaking down chemicals. So we found these 3 compounds and said, well, it's very nice. We see that these chemicals theoretically can bind to the colchicine binding site. They comparably bind with the same sort of energy. Can they actually interact with the microtubule cytoskeleton and influence the way that cytoskeleton polymerizes? So we took those 3 chemical compounds, we found suppliers for them, and then put them into microtubule polymerization assays. And these are the results here. The black curve is our control curve. It has absolutely no chemicals involved. You can see that over time, as the tubulin polymerizes, the density, the optical density in the system increases, and then you see that curve kind of ramp up and then taper off as the tubulin is formed into microtubules. We have a positive control, which is the paclitaxel, which is in red, and then we have a negative control, which is colchicine, and we have various doses of these chemical compounds. So 2 of the chemical compounds, 25B NBF and 25C NBF, are somewhat related with small chemical differences, and we found that additional doses and increasing the dose of that chemical increases the polymerization effect of microtubules, which was kind of we didn't expect that. We expected these, because they were binding at the colchicine binding site, to be destabilizers, but that was not the case. They, in fact, stabilized microtubules. However, the DMB MPP did end up depolymerizing it initially, but as we increased the dose, again, we got to higher optical density. That leads to the question of, is this higher optical density due to more microtubules, or is it due to some sort of aberrant form? Is the tubulin sticking together in clumps? Is it forming some weird sort of sheet or ribbons? And so we went to a biochemical assay and put it under a microscope. In the top 2 panels, we see control and paclitaxel, which shows the microtubule filaments. And on the far right, the destabilizing agent colchicine, you see these little clumps, and that's due to the microtubules being destabilized and depolymerizing. When we put in 25B and 25C NBF, again, we bring back that microtubule filament form. So we are forming microtubules in that case, whereas DMB MPP, as we expect, is actually forming some sort of aberrant clump. So as we get to higher and higher doses, we're just getting bigger and bigger clumps of this of tubulin protein. Now, how does this relate to Arat's work? So Arat had talked to you about photons coming in and interacting with the tryptophan networks within microtubules. That's what's shown here. This is tubulin on the left-hand side with your tryptophan amino acids highlighted in blue. If you strip away the rest of the protein, you see this network of tryptophan within a single tubulin. Now back when I was finishing up my PhD and going into my postdoc, we built a model of energy transfer within tubulin. We did not use the Forster mechanism that Arat was talking about. We used a similar mechanism called coherent resonant energy transfer. Basically, we represented each of the tryptophan as a dipole and then coupled them together by a coupling constant. So it describes how strongly they interact. And we formed a network of how these dipoles could potentially interact with one another and how strongly they would influence one another. So if a photon comes in and excites one of these tryptophans and it sets a dipole oscillating, the way that oscillation affects all of the other tryptophans around it and causes their dipoles to oscillate is mediated by the strength of these connections. We built this and we compared predictions on spectroscopy experiments from the model to experiment, and we found fairly good agreement. So we know that we're at least in the right ballpark, kind of describing this kind of behavior. And then we simulated the dynamics of how this energy could perturb one of these tryptophans and how it would be shared across a tubulin dimer. And this was based on work that was related to energy migration in photosynthetic complexes, which was quite popular back in the early 2010s. Primarily by Greg Engel's group. It was where we based this off of. But we applied it to tubulin and microtubules, and we found a similar sort of effect. We simulated how this energy could transfer along the length of a protofilament, and there's a lot of parameters that we do not know. So we played with parameters and applied different parameter schemes, and depending on which sort of parameters we use, we can get from that energy traveling between 1 and 2 dimers, or it can go all the way up to 12 dimers if we get getting very liberal with that energy transfer, which does not coincide with the findings of Arat's work. So the more conservative estimates do align with the work that Arat has shown. One thing that was interesting is you can see that everything there is shifted to the right. And what that implies is that the energy migration has a preferential direction. It wants to travel from the alpha tubulin in the beta tubulin direction down the microtubule, as opposed to from the beta tubulin side down the alpha tubulin. So there's a directional mode there, at least from these models. Again, this is all completely theoretical. Now, taking these same models, we started looking at how can they affect the way that this energy can be transferred. And so we started docking given chemicals that have those same aromatic ring structures to tubulin, and seeing how it would influence the way that excitonic energy could be transferred. So this is work, this is just some preliminary work that I had done with Jack Tazinski of binding the chemical melatonin to tubulin. And melatonin is that kind of light blue chemical stuck there right beside that tryptophan molecule. The upper panel up there is the way that the energy is shared across tryptophan when melatonin is not involved. And the bottom panel is the way that the energy is shared when melatonin is involved. And I've highlighted those 2 rows. So the green row on top, you'll see that there are 2 gray boxes, and that means that energy, at least for that exciton mode, is shared across 2 tryptophan. Whereas in the bottom, when melatonin is present, it's only shared on 1. And that is one of the major changes that we see, as well as that you'll see a similar mode there when you put in the melatonin. You lose, again, that shared energy between 2 pairs of tryptophan. So what it implies is that melatonin would disrupt this sort of energy transfer process. It doesn't enhance it. What we suspect is if we start to look at how phenylethylamines would interact with this, is that we can use this same formalism and framework to predict whether it would enhance or wash out this sort of optical energy transfer. Now, if this energy migration within microtubules is supposed to play a role in consciousness, one, we would expect that anesthesia would remove it, which is what Arat has already shown. But then we would also expect that on the other end of the spectrum, that things like hallucinogenic compounds would enhance it. We have not shown that yet. We are starting to look into this. We are seeking funding for this. That's my shameless plug. So if you have money laying around, come talk to me later. But besides that, this is something that is rapidly and easily done chemically within a theoretical framework to make predictions. And then working with groups like Arat's group, we can then send them these compounds and they can actually measure it. So this is something that is easily doable and something that we could show in a fairly quick time frame. In summary, microtubules are key structures capable of modulating ion channels, influencing synaptic plasticity within brain cells, and forming that primary structure of neurons. Microtubules are influenced by psychedelic drugs. We've specifically shown that the substituted phenylethylamines can interact with the microtubule cytoskeleton. These phenylethylamines can bind within that colchicine binding pocket near highly polarizable aromatic amino acids. And that the binding of chemicals, not specifically phenylethylamines, but chemicals in general, can bind near these aromatic amino acids and disrupt the way that these aromatics can share energy and transfer energy. These chemicals can then alter the exciton modes, which you would be able to measure experimentally, and further analysis on this is warranted. These are the references that I've referenced throughout there. If you go back through slides, you'll see little reference quotes. These are all the ones that I've been talking about. This work that I'm talking about specifically is online in a preprint. That's reference number 5. So all of this is shown there. And then thank you very much. If you have any questions, I'm here. Also, I'm actively seeking grad students. So please come talk to me if you're looking for a posting.
Can I go work for you? Oh, just kidding. Hey, thank you very much, Travis. We have time for a couple questions. I just want to make a comment about the phenylethylamines enhancing the polymerization. The psychedelics have been referred to as psychoplastogens because they actually promote growth of dendrites and sprouting of neuron, I guess axons too, but dendrites and spines and so forth. So they actually grow new neurons and grow your brain. Maybe not new neurons, but the neurons we have grow more. So psychoplastogens is a new word I just learned, which I like a lot. So any questions for Travis? Cassie.
That was a really interesting talk. I was actually fascinated by the fact that you have these compounds binding in the colchicine binding site, or purportedly, and that they have an opposite effect. That they increase polymerization. So I was curious if you looked at like microtubule dynamicity at all, or if you took those compounds and tried to dock them into other sites, such as the Taxol site or other sites.
Yeah, in this initial study, we only focused docking on the colchicine binding site. And that was primarily, we were just following along the hypothesis of Van Wokerum from back in the 90s. It's something right now that we're looking at further. Tatum and Caleb, my other students, they're starting to look at other sites, such as the Taxol binding site and so forth. I don't have any results because they've only started on it very recently. As far as microtubule dynamicity, maybe we should talk afterward because I'm not quite so familiar with that.
We got none here. And then anybody else?
I was just curious about the different phenethylamines that you guys were testing, because I saw on the list of, if you're looking at the different Shulgin analogs, there was a lot on that list of the NBOMEs, and that those can be, if we're getting into sort of human testing or that type of phenomenon, that those can be essentially like structurally unsafe. In different ways. So I was just curious if that was the whole list of things that you had tested, or if you had specifically sort of chosen that classification of Shulgin analogs to look at in reference to your other research.
So the ones that we have there are NBOMEs within that list. I don't know if they were within the top 10% of compounds there. We only selected compounds which were known to have a psychedelic effect. We did not screen for safety. So part of this work has been looking at, one, what is the effect of any of these substituted phenylethylamines, and whether they could be beneficial, as in what Stuart was talking about being a psychoplastogen, promoting microtubule stabilization, which may be used in illnesses such as any sort of neurodegenerative disease, or if they could actually, work the opposite way, which we saw with that DMB-MPP compound, which would destabilize microtubules, and you would get a withering effect of your neuron, which is not something I would recommend. But it is something we're trying to consider. Yeah. Yes.
I think there's some good mechanistic evidence that suggests that there's actually dedicated receptors that support hallucination due to some of these compounds that you've mentioned, NMDA receptors or serotonin receptors, right? So are you imagining that this potentiates those effects,
yes.
or you imagine it replaces them?
No. At no point am I saying that all the large body of literature that states that the specific membrane receptors are involved within these experiences. I'm saying that this would either add, enhance, or modulate. It's been shown that all membrane receptors are, well, not all, but they're anchored within the membrane by microtubules. So if you're modifying the microtubules, you can affect the way that those membrane receptors are responding. Whether that is through direct modulation of the microtubule cytoskeleton, or also interacting with the chemical interacting with the membrane receptor itself. So I think that there's dual roles here going on. I don't think I think it's well known now that chemicals are not specific. They're kind of like a blanket effect. They have multiple targets. They're very promiscuous. And so the rich difference in psychedelic experience you may get from chemicals that are very, very close could be because they're targeting multiple different sites in the way that they act with them differently.
Can I make a comment on that before Dr. Friedman? And that is that I always thought that the psychedelics might get in the cell and bind directly to microtubules because they're nonpolar and hydrophobic. But I was always stumped by the fact that ketanserin, the 5-HT2A blocker, blocked the psychedelic effects. But if the 5-HT2A receptors are inside the cell, sitting in the microtubule-associated proteins, that would explain why how the psychedelics can act pretty much directly, but also be blocked by ketanserin.
Yeah, very nice talk. What you're isolating the effects on the microtubules, but if a person ingests these, they do other things like blood flow. For example, you didn't talk about psilocybin, but psilocybin reduces blood flow in the medial frontal lobe, and that can actually reduce self-awareness. So when you translate it into effect on blood flow and perhaps other things, you might get different kinds of effects. Can you comment on that?
Yeah, it goes back to my previous comment, is like these chemicals are targeting many different systems. I'm looking at the microtubule cytoskeleton because that's my area of research. I'm not saying that it's the prime site of psychedelic experience. It may be, and I know that's what Stuart would think, but reducing blood flow, interacting with the serotonergic system, those are contributing in some way to the psychedelic experience. I specifically am only looking at microtubules, in this case, microtubules alone. It's not even looking at microtubules within a cell. Currently, that's something that we're running. We have neuroprogenitor cells that are being grown with these chemicals to see how they're influenced by these chemicals. But it's kind of a bottom-up approach, whereas other psychedelic research is looking from kind of top-down, looking at whole brain, fMRI images and so forth. So I'm not ruling out any of those effects whatsoever in this case.
I would just say that reduced blood flow is an effect rather than the cause of mental changes. So let's thank Travis for a fantastic talk.